My name is Sara, and today I will be talking about better understanding the diversity and functions of the skin microbiome using genome-resolved metagenomics. So I am an MD PhD student interested in going into dermatology, and for the PhD portion of my studies, I was able to design a collaborative project between two very innovative labs. So this is the Fin group at EMBL EBI, which is in Kingston, UK, and the SIG-RAY lab at the NIH NHGRI, which is in the Maryland in the US. So for my PhD, I wanted to focus on the microbiome. The microbiome is the set of all bacteria, viruses, archaea, and eukaryotes living in and on our bodies. And the microbiome is vast, so it's on the scale of 10 to 100 trillion microbial cells. So what has always intrigued me is that if we actually quantify the number of cells in our microbiome, we find that we are as much microbe as we are human. In addition, while we are 99.9% identical to each other, we are roughly 80 to 90% different from each other with respect to our microbiomes. And the gut microbiome is currently a very hot area. I think we have all heard of studies that have associated the gut microbiome to various conditions such as obesity, autoimmune diseases such as inflammatory bowel disease, and even how well patients respond to chemotherapy. And to understand how variations in the microbiome modulate disease, we first need to know what microbes are present in health and how they shift in various disease states. So there are two main ways of answering this question. So the traditional way is culturing, and then we can sequence the cultured organism. Examples of this work include the culture collection by the Fin group and the Lolly labs. However, these collections are limited to what can be grown under standard laboratory conditions. So alternatively, we can use sequencing to get the genetic information about all microbes present in a sample. So this is called a metagenome, and we can reconstruct the microbial genomes that constitute the community and using the sequencing reads themselves. So examples of this work included the work of the Fin group and collaborators, where they analyzed roughly 10,000 metagenomes and found roughly 2,000 novel bacterial species. So I was really intrigued by these approaches and wanted to apply these methods to the skin microbiome, which I find to be just as exciting as the gut microbiome. Before I talk about the skin microbiome, I first wanted to discuss our skin. So our skin is not the same all over. We know that, for example, our face is oily. So our elbow creases are moist. Our forearm is dry. And this is important because these different microenvironments, so oily, moist, and dry, they tend to harbor different microbes. So sebaceous sites are dominated by the lipophilic Corynebacterium species, while moist sites are dominated by Staphylococcus and Corynebacterium species. So these are microbes that really thrive under humid conditions. And why is this body site specificity of skin microbiome microbes important? Because skin diseases with hypothesized microbial basis, like those listed in this slide, tend to affect specific body sites. So here we have atopic dermatitis, or otherwise known as eczema, which affects roughly 15 to 20% of children worldwide. And it has been associated with the overgrowth of the bacterium Staph aureus, and it typically presents in the elbow pit and behind the knees. In contrast, psoriasis affects things in an extensor distribution, such as the knees and the elbows. And seborrheic dermatitis, which has been associated with Malassezia species, tends to affect the scalp. We talked about how certain microbes prefer certain body sites. So one could ask whether the site specificity of these microbiome-associated diseases is a consequence of the body site tropism of the disease-causing microbes themselves. To answer this question, we need to know what constitutes the microbial composition of these body sites during health and disease. The previous work at the Spicere lab had answered this question using shotgun metagenomic sequencing. So what they did is that they took 12 healthy volunteers and sampled various body sites of these volunteers, and then took the sequencing reads that were generated, and then compared each sequencing read to the genomes of already cultured organisms. So these are things that you can find in RefSeq or GenBank. And these are read classification-based methods. And what they found is that roughly half of the reads did not match the genomes of cultured organisms. So to circumvent the issue of incomplete databases, what we decided to do is we decided to culture from the skin of these healthy volunteers. And although culture-based approaches yield high-quality genomes that enable farther mechanistic insights, again, they are biased towards members of the skin microbial community that can be cultured under laboratory conditions. So to provide a more comprehensive survey of the skin microbial community, we also decided to use sequence-based methods such as de novo assembly and binning of the shotgun metagenomic read. So approaches used by the Finn group in the gut. So we decided to analyze all publicly available skin metagenomes, but we also did a focus study of, again, these 12 healthy volunteers that had been previously studied by Julie's lab. So these healthy volunteers were brought back to the NIH and they were sampled for this 4th time point. And we had deep sequencing of different body sites of these healthy volunteers for these 4th time points. And the resulting metagenomic data set was of 12 healthy volunteers, 19 body sites, and up to between 3 to 4 time points for each of these volunteers. So once we have these samples, what did we do with them? So again, Heidi took these, Heidi's the clinician at the NIH, and she took these samples from these healthy volunteers. And then these swabs were, the material of these swabs were sent for sequencing, and we ended up with short reads. So this is the output of the Illumina sequencing. Then we performed de novo assembly of these shotgun metagenomic reads into longer contiguous sequences via metaspades. So assembling short reads into contigs has many advantages. So longer stretches of sequence are more informative about what organism the sequencing reads belong to and get us closer to reconstructing these organisms' genomes. But as you might expect, it's super rare to reconstruct an entire microbial genome from a short read assembly. So what we can do to get us closer to reconstructing the whole genome of these organisms is that we can group contigs based on similarities in sequence properties. So things like tetranucleotide frequency. And what we can also do is that we can do read recruitment and then look at the depth of coverage of these contigs across related samples. So this is the idea being that if 2 contigs belong to the same organism, the depth of coverage across different samples would be the same. So the output of this step is bins. And again, so these are groups of similar contigs. And these bins can undergo bin refinement and also quality filtering. So there are these criteria for mags that were proposed, and they're called MyMag criteria that set quality requirements for including a bin And so what we end up with is metagenome-assembled genomes, or MAGs. So oftentimes we do a lot of our studies at a large scale, and we end up getting the same microbe as a part of multiple studies. So to get an idea of how many distinct species these MAGs represent, we can dereplicate, or in other words, reduce genetic redundancy of our set of genomes to obtain a set of species. So this basically involves looking at the genetic similarity between sets of MAGs and choosing one representative for each set of species cluster that are found in our data set. So a challenge with using techniques previously used in the gut is that the skin is a low biomass site. So when we get our samples, you have a lot of human DNA. And once we remove the human DNA, we actually end up with very little microbial reads. So what we did is we analyzed the sequencing reads at the level of a single sequencing run or a single sample, but we additionally pooled samples across different metadata variables. And this is otherwise known as co-assembly, or what we call here as pooling samples. So in the case of pool healthy volunteer, for example, we aggregated all the sequencing reads of all body sites of this one healthy volunteer, and then put that as through our pipeline. And in the case of pool site, we took the same body site of many different healthy volunteers and aggregated those reads, and again, put that through our pipeline. We also tried another approach, which is pool time, where we took the same body site of the same healthy volunteer across multiple time points and then aggregated the sequencing reads across those multiple time points. And these were our different pooling strategies. So in addition to these metagenomic analyses, so Clay had cultured from body sites of seven of these healthy volunteers. And as a result, these were all done with varying lipid and oxygen levels to sort of increase the diversity of microbes that could be cultured. And he supplemented this culturing with microbes that were isolated from patients with skin disorders that have been shown to change the diversity of the skin microbiome. And what we ended up with is 251 isolates that constitute the skin bacterial culture collection. And here I'm just showing some of the isolates that he was able to recover from different body sites of just one healthy volunteer. So to assess the quality of our MAGs, we then compared our MAGs to their matching isolate genome. So here I'm showing a MAG and an isolate, and we would expect that MAGs are incomplete. And so here, that's why I'm showcasing the MAGs as being smaller than the isolate genome. And then we looked at MAGs and isolates that matched at a species level. And what we defined as a species level match is whether they aligned at least 30% with at least 95% ANI. So this was a criterion that was proposed by Alex in the Fin group in his Nature Biotech paper. And we took all of the MAG isolate matches, and then we calculated metrics such as MAG aligned, which is the length of the alignment divided by the genome size of the MAG, and the isolate aligned, which again is the length aligned divided by the genome size of the isolate. And here I'm showing a plot of the percent MAG aligned and the percent isolate aligned. And again, as we mentioned, MAGs are incomplete, so their genome size would be smaller, and this would result in the percent MAG aligned would be expected to be higher than the percent of the isolate aligned. And if we draw this identity line, we see that as expected, all the points lie above this line, signifying that the percent of MAG aligned was higher than the percent of isolate aligned, which was reassuring because sometimes we worry about the generation of chimeric genomes, where in those cases, perhaps this would not be the case. We also looked at how the isolate genome correlated, the isolate aligned correlated with the CheckHim completeness. So I will first describe how CheckHim works. So when we try to assess the quality of a MAG, we often don't have any representative genome for that MAG. So what is done to approximate the quote-unquote completeness and contamination of these MAGs is by looking at single copy marker genes. So these are genes that have been found to be present in all prokaryotes in single copy. And so let's say that there are 10 single copy marker genes. If we find 9 out of the 10 single copy marker genes in our MAG, we can approximate that the MAG is roughly 90% complete. But if we find one of those single copy marker genes twice, then we can approximate that perhaps the MAG is 10% contaminated. And here we find that as we go towards increasing isolate aligned, the CheckHim completeness also increases. So they tend to correlate with one another, which is again reassuring. But we had done some farther analysis, which showed that actually CheckHim tends to overestimate the completeness of MAGs compared to what we had found in our alignment approaches. So we also see in this figure that the pooled MAGs, which are shown in this triangle, tend to have a high isolate aligned and MAG aligned, and also tend to have a high CheckHim completeness. And in fact, when we looked at these metrics of MAG aligned and isolate aligned, and then subsetted them by pooling strategy, we found that with increasing sequencing depth, so if we arrange the x-axis in order of increasing number of reads or sequencing depth, we get a higher percent of MAG aligned and isolate aligned, suggesting that these pooling strategies really help us generate higher quality genomes. And here I'm just showcasing a single, like a Corynebacterium species that was recovered as both an isolate and a MAG in our studies. And this is a novel genome, as in it is substantially different genetically from what is found in public repositories. And the MAG was recovered only via pooling across 86 different sequencing runs from different body sites of one healthy volunteer, so the pool HV strategy. And in this plot, we see the isolate was also recovered from the same healthy volunteer at the same time point. And in this plot, we see the alignment between the query, which is the MAG, and the reference, which is the isolate genome. And the ideal scenario would be a 45-degree line that would signify a perfect alignment between the MAG and the isolate. But in this case, we see a portion of the isolate is missing from the MAG, but overall, we had 99.9% of the MAG aligning, 83.3% of the isolate aligning, with 99.9% average nucleotide identity. And this high correlation between the MAG and isolate genomes really showcases how powerful these de novo assembly and binning approaches are in reconstructing genomes from metagenomes. So we found that our pooling strategies allow us to generate high quality MAGs in terms of both the CheckHim completeness and also after quality assessment with isolates. But do they allow us to capture additional diversity? So I took the 10,000 roughly MAGs that we had generated and dereplicated them. So this is, again, clustering them based on genetic identity into species level clusters. And then I tried to see how many of these pooling strategies were able to recover each of these species. And so I'm boxing so this is an upset plot where I'm boxing the top 7 strategies where we recovered the most number of distinct species. And here we find that while much of the MAGs were recovered by the majority of the pooling strategies, a subset of the MAGs were unique to only a handful of the pooling strategies. So for example, the pool HV and the pool HV and the pool site. And this really shows that there's really a benefit to each of these strategies that we had tried, as perhaps each may preferentially enrich for a microbe and allow it to be recovered as a MAG. So now I'm showing a phylogenetic tree of the 622 dereplicated bacterial species that make up the SMGC. So this outer ring is the taxonomic classification of these MAGs and islets. So just to clarify, so in the SMGC, if a MAG and islet represented the same species, when we dereplicated, the islet genome was preferentially chosen over the MAG. So here when we see islet and MAG, it doesn't necessarily mean that we did not recover a MAG for this islet. It's just that the islet genome was preferentially chosen. But what we are showing here is that the novel genomes, so this is either a novel MAG or a novel islet, which are shown in red or this bright green color, they sort of are interweaved with the known. So this is the blue and the gray. And what this shows us is that perhaps some of the reasons that these genomes are missing from public repositories is just insufficient culturing. That with greater culturing, perhaps we will have representatives for all of these genomes that are missing. And here I am showing the relative abundance of the top most abundant genera on the skin. And this is in contrast to previous surveys that had underestimated, for example, Lawsonella. And we also uncovered this abundant novel genus, which we named Pelibacter. And we can see that it's abundant, it's primarily found in sebaceous sites, such as the alar crease or the forehead or the cheeks. This all suggests that perhaps this genus is lipophilic. And to understand again why we had not cultured these bacterial species before, we looked at the number of species and how many healthy volunteers they were detected in. So the majority of the species were only found, so we have this U-shaped distribution where the majority were found in only a few individuals, but the novel species were spread throughout this distribution, suggesting that they're not just unique to a few individuals, and that these are not just low abundance or transients. We even had examples of species that were found in all healthy volunteers. So we just discussed the bacterial side of things. So next I'm going to go to the eukaryotic side. So this was all made possible by UCC, which is the equivalent of CheckM, but for eukaryotes, and was developed by Paul Sari, who is another pre-doc at the Fin group. And eukaryotes on the skin include things ranging from fungi such as Malassezia to mites. And Malassezia species are particularly interesting because they have been associated with both healthy human skin, but also various cutaneous disorders. So characterizing their diversity is particularly important. So we got these eukaryotic mags, and then for taxonomic assignment of these mags, we first performed whole genome alignment against fungal genomes from publicly available databases, so like RefSeq and GenBank. And based on our whole genome alignment, we found 3 novel Malassezia species. So we also recovered what have been reported for the skin, so Malassezia globosa, restricta, and sympodialis. And to better understand the phylogenetic positioning of these 3 novel Malassezia species, we generated a tree for our mags and also reference genomes. So our mags are shown in blue and the reference genomes are shown in red. So again, we find our Malassezia restricta, globosa, sympodialis, but these 3 Malassezia species that really don't have any representative in publicly available databases. And then when we look at the prevalence of these Malassezia species, we find that Malassezia globosa and restricta were detected in all healthy volunteers and in multiple body sites. But the novel Malassezia species fall into 3 classes. So the first class is ubiquitous across all healthy volunteers and many body sites. The second class is found in a few healthy volunteers and in many of those body sites of those healthy volunteers. And the third class is only found in 1 healthy volunteer and in a few body sites of that healthy volunteer. So in contrast to these, so Malassezia species, although these were novel, they were actually found in multiple time points of these healthy volunteers. So they were relatively stable over time. In contrast, we detected potential transients like Deboromyces and Clavispora, which were found at a single time point and individual, and were often detected in what we call high diversity sites. So things like our hands and feet that are frequently exposed to the environment. So next we looked at the viruses. So for this analysis, I used Verify, which was a tool developed by Martin at the Fin group. And this allows us to input our metaspate assemblies, and then it runs these assemblies through VerFinder, VerSorter, and PPR meta. And then it determines some putative viral sequences that undergo quality control, so they can, because we often worry about false positives with viral analyses. One way to reduce false positives is by length filtering. So we removed all viral sequences that were shorter than 5,000 base pairs. We also consulted CheckV, which is a tool for quality assessment of viral sequences. So obviously we don't have single copy marker genes for viruses. Instead, CheckV identifies non-virus components, so what we call contaminants, by looking at gene annotation and GC content. And it also estimates completeness based on comparison with a large database of complete viral genomes that were derived from NCBI GenBank and also from environmental samples. So I clustered the SMGC viral catalog, and I ended up with a set of 15,591 prokaryotic and eukaryotic viral genomes. And what was interesting to me is that the majority, so roughly 90% of our viral sequences did not have any match to what was found in IMGVR. So to understand what these viral sequences belong to, I clustered them by protein content with the RefSeq prokaryotic sequences. So here we have the RefSeq prokaryotes in light blue, and the SMGC is, you see like a big clustering with the RefSeq prokaryotic viral sequences. So this big cluster belongs to Cauda, which includes the families Myoviridae and Cyphiviridae. We also recover eukaryotic viral sequences that have been previously found on human skin. So these include Papillomaviridae and Poxviridae. But in addition to all of this, we also recovered jumbophages. So these jumbophages are prokaryotic viral genomes that are longer than 200 kb. And they're of particular interest, not only due to their large size and uncharacterized genetic diversity, but also as potential agents of phage-based therapies. And we detected 20 distinct jumbophages, which the maximum was 380 kb in size. And these formed roughly 5 major clusters. Our cluster 5 jumbophage with size of roughly, I think, 260 kb was in the same cluster as an Acinetobacter jumbophage, which had been previously isolated from a wastewater effluent and had been reported to be able to lyse the clinically important nosocomial pathogen Acinetobacter baumannii. And this phage was present predominantly in the foot sites, where we also found the Acinetobacter baumannii MAG present. Looking at the distribution of these jumbophages, we find that they were found in multiple individuals. And again, they were found in these high diversity body sites like the hands and the feet. So overall, viruses are important components of the microbiome, and they play important roles in modulating the diversity and functions of their hosts. So our work provides an important resource for future investigations into the skin microbial dynamics. So we also looked at how well the SMGC classified the skin metagenomic reads compared to 2 different databases. So this includes the Pozzoli skin and the standard Kraken database. And what we found is that the SMGC not only increased the median of the reads assigned, but it also reduced the variance in the percent of reads assigned. And we recovered some of the highest reads assigned in the oily body sites, but instead in the high diversity sites like our hands and feet, we still have a lot of work left. So these sites, we believe, are frequently exposed to the environment, and even with the robust pooling strategies that we used in this work, I don't think we were able to achieve sufficient sequencing depth to recover these genomes as MAGs. And in support of this theory, we found that when comparing the composition of the same body site of an individual healthy volunteer across multiple time points, we found that in the sebaceous sites, the theta dissimilarity was low. So in other words, the sebaceous sites, their composition is relatively stable over time, but these high diversity sites like our hands and feet tend to vary dramatically between different time points of the same body site of the same individual. So this could be one of the reasons that it makes it really hard for us to incorporate these genomes into our catalog. So in terms of accessing the SMGC, so currently the SMGC is in an FTP link, but in the coming weeks, we plan to move it to Magnify, which is also where the UHGG is housed. And the conclusions are that we generated the skin microbial genome collection, which spans roughly 620 prokaryotic species and provides an increase of 26% in skin bacterial diversity. We also recovered some novel eukaryotic species and thousands of viral sequences, including clades of jumbophages that were prevalent in foot sites. And SMGC now allows us to classify roughly 86% of the skin metagenomic sequences, and provided us with a new map of what is found on our skins, which I thought was very exciting. Thank you so much for taking the time to listen to this talk.